And welcome back to Med School Radio, where we play your favorite hits. Okay, we're going to be talking about rheumatoid arthritis, and this time we're going to be talking about the B lymphocytes. As B cells are generated in the bone marrow, in the peripheral blood, B cells are greatly outnumbered by T cells, constituting 10 to 15% of the lymphocyte population. In the spleen, however, B cells represent one half of the lymphocytes present, emphasizing the importance of mounting an antibody response to foreign pathogens and antigens. B cells express on their surface membrane-tethered immunoglobulin molecules that function as B cell receptors or BCRs. These receptors recognize a much larger peptide structure termed the antigenic determinant or epitope than do TCRs. Thus, B cells do not, like T cells, require processing of antigen into smaller peptide fragments embedded into the surface of MHC class 1 or 2 molecules. What accounts for the difference? B cells are involved in policing the bloodstream and body fluids and therefore have immediate access to foreign antigen. T cells, in contrast, are charged with unmasking a hidden invader, a virus, for example, that lurks inside a cell. B cells also express a surface receptor for the fragment crystallizable Fc region on the IgG molecule, as well as receptors for activated complement pathway intermediates. In addition, B cells can act as APCs for T cells. The Fc portion of the immunoglobulins carries out its effector functions such as binding complement fragments and opsonizing bacteria to make them more palatable to neutrophils and monocytes. Now let's talk about B cell development. There are two phases to B lymphocyte development as follows: an antigen independent early phase and an antigen dependent later phase. Say again, there are two phases to B lymphocyte development: an antigen independent early phase and an antigen dependent later phase. The antigen independent phase occurs in the bone marrow, while the antigen dependent phase occurs in germinal centers of the secondary lymphoid organs such as the spleen and lymph nodes. In the antigen independent phase, a stem cell undergoes a series of maturational stages that include ordered expression of genes, expression of a succession of cell surface markers, and rearrangement of the immunoglobulin gene. This stage generates a pre-immune or inactive antibody repertoire of naive B cells. During the second antigen-dependent phase, this primary repertoire of antibodies is greatly diversified through a process of antigen interaction. As a result of this refining process, antigen specificity is greatly enhanced, and two populations of cells are formed: memory B cells and antibody-secreting plasma cells. Diversity of the antigen-binding variable region of the immunoglobulin is generated by heavy and light chain rearrangements, basically involving shuffling of variable V, diversity D, and junctional J gene segments. Thus, the enormous diversity of immunoglobulins with different epitope specificities is created by varying the pieces of DNA used in the manner in which they are combined. Antibodies are Y-shaped molecules, with the stem of the Y being the constant region and the arms of the Y bearing the hypervariable or complementarity determining region, the sites where the antigen binds to the antibody. In addition to varying the gene segments and the ways the gene segments are combined, a process termed somatic hypermutation occurs in the hypervariable region, whereby point mutations produce mutant sig molecules, some of which bind antigen better than the original SIG molecules, or SLG molecules. The randomization of genes is also likely to generate self-reactive antibodies. To correct this aberration, autoreactive BCRs can be mutated so that they no longer react with self-antigens. If this editing fails, such cells undergo negative selection in the bone marrow and are disposed of by apoptosis. Now let's discuss immunoglobulins, humoral mediators of acquired immunity. Antibodies afford protection against many pathogens through an ability to bind specifically to an antigenic determinant complementary in structure to that of the antibody. This specific binding marks the microbe so that it is made more susceptible to the phagocytic action of neutrophils. In addition, antibody binding functions as a trigger to initiate the set of enzymatic reactions known as the complement cascade. Antibodies are Y-shaped glycoproteins composed of paired identical heavy and light chains joined by several covalent disulfide bonds. Located within the variable region is the complementarity determining region, which is where the antigen binding occurs. The constant region, Fc region, binds complement, triggering activation of that system, or binds to cell surface receptors or monocytes or macrophages. It should be clear that with immunoglobulins, the framework of these molecules is conserved to provide a stable structure upon which to deploy a unique antigen binding site, or FAB region.